So let's take a look at 2.25. So this is a problem that a lot of students had trouble with on the exam. So the question is, the nuclear DNA content of a single sperm cell in Drosophila melanogaster is approximately 0.18 picogram. What would be the expected nuclear DNA content of a primary spermatocyte in Drosophila? So given the information that a sperm cell has 0.18 picogram, how much DNA is going to be in a primary spermatocyte? So I think the easiest thing to do here is to draw spermatogenesis out, map it out again. Primary spermatocyte goes to secondary spermatocyte, two secondary spermatocytes, goes to 4 spermatids. Now, this is meiosis 1 right here. This is meiosis 2. And it might help to think of the chromosomes, what's going on with the chromosomes at each of these stages. So I'm going to draw a tetrad here, just one. I think Drosophila has 4 chromosomes, so there would be 4 tetrads here. I might be wrong on that, but I just want to map out one tetrad for you. So this is one tetrad, one of the 4, and so during meiosis 1, well, the tetrad's going to split, right? One dyad's going to go to one secondary spermatocyte, and another one, I'll just draw this one side here, is going to go to the other secondary spermatocyte. And during meiosis 2, well, then the centromeres are going to split. So we have a monad going this way, one going here, one going here, and one going here. So these spermatids, the question tells us that they have 0.18 picogram of DNA. So you can see that the secondary spermatocytes, well, they have twice as much DNA as the spermatids. So these should have 0.36 picogram of DNA. And the question is, it wants the amount of DNA in the primary spermatocyte. So, and you can see by looking at the chromosomes that the primary spermatocyte is going to have twice as much as the secondary spermatocyte. So this is going to be 0.72 picogram of DNA. So that's the answer to the first part of the question. 0.72 picograms of DNA is the amount of DNA in the primary spermatocyte. So the next part of the question is, the next question is, what would be the expected nuclear DNA content of a somatic cell, which is just not a gamete, not a sex cell. What would be the nuclear DNA content of a somatic cell in the G1 phase? So I think the easiest way to look at that is, we already know, well, recall that spermatids are haploid, right? They have one set of chromosomes. And if spermatids are haploid, and haploid cells have 0.18 picogram of DNA, and Drosophila is a diploid organism, so in the G1 phase, well, we're just going to have, you know, instead of being haploid, we're diploid. We're just going to be a diploid cell. We're going to have 2 sets of chromosomes. So Let me clarify that up here. So here I have represented the DNA in a spermatid, which is haploid, and now I'm representing the DNA in a somatic cell, diploid cell, in the G1 phase. So there's just twice as much DNA there. So the answer to that one is going to be 0.36 picogram. And now the next question is, what would be the expected nuclear DNA content of a somatic cell at metaphase? And I think the easiest way to look at this is to consider what happens right after G1, right? As long as the cell isn't going into G0, it's going to S phase. Okay, and during S phase, the DNA is replicated. So I'm going to make the sister chromatids here attached to common centromere. So we still have, just 2 chromosomes depicted here, but now we have the sister chromatids attached after S phase. And after S phase is finished, well, that's what the cell is going to look like in G2. And I could probably just copy and paste that. Okay, so this is G2, which is the same. And now the cell's ready to go into mitosis. And so during mitosis, these chromosomes are going to make their way to the metaphase plate. After going through prophase and prometaphase. Okay, and we have the metaphase plate here, and they're aligned, and this nuclear envelope has gone away. They're attached to the microtubules. And okay, so the question again, what would be the expected nuclear DNA content of a somatic cell at metaphase? Okay, so go back up here. Well, we already determined that the amount of DNA in the G1 phase was 0.36 picograms. So after S phase is finished, well, we've doubled the amount of DNA. So we have 0.72 picograms. So 0.72 picograms in G2, and we don't double the amount or reduce the amount of DNA after that point. All the way up to metaphase, we still have 0.72 picograms of DNA. And then when anaphase starts, well, then half the DNA is going that way and half is going that way. Okay, but the question wants to know how much DNA is in the metaphase, in the cell during metaphase. So 0.72 picograms is the answer.